Okay, we're going to move on to our next session that's starting. That's fine, yeah. Can I relax while I'm noticing? Yeah. I'll try to keep it short. Okay, so this is our basic science plenary lecture. And we have joining us today Dr. Laura Odell. And so Laura is currently an Associate Vice President for Research and Professor of Behavioral Neuroscience at the University of Texas, El Paso. She received her PhD from the University of Arizona with Janet Nieswander, and then she did her postdoc and staff scientist positions at the Scripps Research Institute with George Koob, who's now director of NIAAA. She's had a very successful career in science. She has many really impactful publications and findings that have really shaped the field. And she's also been at the cutting edge of a lot of integrative kind of ways of thinking about nicotine and nicotine's actions, not only in the brain, but in other bodily systems. So in 2008, she was a Presidential Early Career Award for Scientists and Engineers, or PEACE Award winner, and that's from the National Science and Technology Council, which is actually the highest honor bestowed by the U.S. government for outstanding scientists beginning their independent careers. More recently, in 2017, she received the Research Exemplar Award, which recognizes individuals who conduct high-quality, high-impact research and exemplify the professionalism and integrity in research. Within her research, she does use rodent models to study the underlying neural mechanisms that promote substance misuse in vulnerable populations, including females, adolescents, as well as individuals with diabetes. Her laboratory combines both neurochemical and molecular approaches with behavioral models to provide a better understanding of the mechanisms that promote drug abuse with a particular focus on nicotine. Now, although her lab does focus on nicotine, I do have to say that there's a lot of interest in e-cigarettes and e-cigarette vape, and Laura was one of the first to look at vape exposure, although she started with alcohol in doing those studies. So she has a long history of vape, so all of you that are trying to implement vape studies, she's our expert here. So she also was one of the first to start looking at sex differences between males and females, and she's really an early leader in this direction. She's one of the first to start looking at other comorbid conditions, so with a focus on diabetes, so using insulin and different diabetic rat models. Again, among the first to investigate this connection. She's also looked at food take, obesity, and high-fat intake, and how that intersects with nicotine use and abuse. And then going down to the molecular level, looking at differences between nicotinic receptors and serotonin receptors and opioid receptors. So really a lot of really early studies, and then continuing that into more of an understanding for us to understand how there's these intersectionalities or comorbidities that are really important and continue to be really relatively underexplored, and we have a lot to still learn. For the past 20 years, her research program has been funded by the National Institute on Drug Abuse and other organizations such as the American Diabetes Association. And as a final little plug for her, Dr. Odell really has a deep interest in promoting young faculty and trainees who want to pursue a career in neuroscience, and she's really supportive of those from diverse and minoritized backgrounds. And she's received multiple awards recognizing her exceptional mentorship of trainees. And I think as a PI, seeing your trainees go off and do amazing things is one of the most rewarding things of our career. So being really able to provide that mentorship is really an exceptional quality. Indeed, she also leads a summer mentoring and research training program, which has science education and research training for undergraduates and high school teachers with a focus on the neuroscience of drug addiction. And with this focus in mind on using science as a platform for promoting diversity, she's given talks at various groups, including NIAAA, NIDA, and NIMH. And so it's with great honor that I welcome Dr. Odell to the stage, and I really look forward to your talk.
Thank you, Christy. I'm going to give you like a little hug. Thank you. Wow, I always say I wish my mom was here. But I just want to say thank you, Christy. I'm a huge admirer and now just pleasure to call you a friend. So thank you for that really generous introduction. Thank you for bringing me back to SRNT. It's been really fun, and I guess on the stage I'll commit to being a better steward to this organization, building up the basic science components of SRNT. And so I'll start off with a little bit of an introduction about myself. Christy already covered this about me, but I'm very, very interested in using my science as a platform to promote diversity. And so this is one of the things I like to say about myself, especially when there's young trainees in the crowd, that I'm somebody that can serve as an advocate and mentor. And it's been really nice to serve as a faculty member at UTEP, where my personal mission is very closely tied to the mission of my institution, and that is to use research as a platform to elevate and promote young trainees. And so that is a picture of me as an undergraduate. I actually went to Texas A&M, but this is me as a faculty member. So what a gift. And I've had a lot of wonderful mentors that have supported me along the way, including Janet Nicewander and George Koob, as you mentioned. And so I really like to think about diversity in terms of the work that I do at an individual level, working with individual students that have unique and special challenges and talents. But I also like to think of my work in a scientific community, but also at the institutional level, where I'm working with the NIAAA on their advisory council to try to promote different platforms where we can promote diversity. And so I do have a little honey of an undergraduate program called the Smart Mind, which I would love to receive your students. We'll take good care of them, and we spend an intensive summer working on the problems related to nicotine use. And so UTEP it was a place where I could really stop and explore why it is that different groups of people appear to be more vulnerable to using tobacco. And I'll start out by saying I'm a rodent researcher, trying very hard to make sure that all of my work has clinical implications. And so these two diagrams were drawn by an undergraduate who I later learned is a tattoo artist, and I don't have this tattooed on my body, but I use it a lot to make the point. I had some stick figure drawings on the board, and she said, "Can I draw those for you?" And this is what she came up with. And the first diagram is really just showing, it's a funnel, and showing nicotine's effects in the brain. And what I've really carved out for myself is a focus really on studying why these different groups are more vulnerable. And so it has diabetes, persons with mental health challenges, adolescents, and females. And so today I'll spend some time giving you a little taste of those different arms of my research program. Now, obviously with rodents, we have a lot of limitations in terms of the different groups that we can model. But the way that I look at my program is looking at two sides of tobacco use, which is, first of all, its pleasurable effects. So we focus a lot of our time looking at the short-term rewarding effects of drugs of abuse. But we also spend significant effort looking at withdrawal. So following chronic use, withdrawal symptoms that emerge that could lead somebody to relapse or even motivate continued use. And so three sort of major points I want to make is that we look at group differences. We combine behavioral measures with different biological markers. And so as we marry those things in different types of animals, there's many challenges, and I'll touch on when I go to each of those groups. But this is a silly cartoon, but it's really what we're trying to do, which is model nicotine use in rodent models. And so we've developed rodent models of metabolic disorders, like diabetes. We also study adolescents and females. And on the reward side, we use conditioned place preference, where the animals are basically pressing a lever to receive infusions of the drug. And we also measure the amount of time animals spend in environmental compartments paired with nicotine. For withdrawal, for a long time we used the nicotine pump, which delivers continuous administration of nicotine subcutaneously. But more recently, we've been studying withdrawal after an animal has been exposed to chronic nicotine vapor. And so we can either remove the animals out of vapor or give them a nicotinic receptor antagonist to precipitate a withdrawal state. And on the biological side, there's an array of different biological measures that we take. My talk today will talk a little bit about changes in gene expression using PCR methods that allow us to target very specific nicotinic receptor subtypes. And we also use neurochemical methods that pair in vivo microdialysis with mass spec procedures. So today, as I mentioned, I'm going to cover 3 arms of my research program, which is focused on adolescents, females, and persons with metabolic disorders such as diabetes. And so I'm just going to start out by making a few points about why we should care about these different populations and the vulnerabilities that exist in a clinical setting. So adolescent nicotine use, as we know, and we heard wonderfully from the previous speaker, Jennifer, I don't know if she's here, but what a great talk, amazing young scientist. But there's a lot of data showing that e-cigarettes have accidentally or unintentionally increased the use of e-cigarettes, including new users. And there's a lot of adolescents that are using nicotine, and the data suggests that if you start during adolescence, you're more likely to continue using as an adult, particularly if you're a female. And so as a result, this means that adolescents are more vulnerable to the long-term effects of chronic nicotine exposure. And so this is work that we started quite some time ago, and back in 2007, we've learned a lot. But at that time, the mechanisms that promoted nicotine use in adolescents were largely unexplored. And so I'll just take you a little bit back in time, where we studied adolescents in rats. And so there's some debate in different ways in which you can differentiate the adolescent period. But in the studies I'll show you, the animals are tested between postnatal day 28 and 45, and the adult animals are over 60 days old. And so on the left, what I'm showing you is a condition place preference procedure where we first test what side of a compartment the animals want to spend time on. And we take the compartment that they do not prefer and we repeatedly give them injections of nicotine. And so the idea is that over time, they associate those environmental cues with the rewarding effects of the drug. And the way that I present that data is a different score. So it's the amount of time they spend after conditioning minus before conditioning. So all you really need to focus on is so that values above 0 reflect the rewarding effects of drugs, and values below 0 reflect aversive qualities of those doses of nicotine. And so what I'll first point out, so these were different doses of nicotine in separate groups of adult and adolescent animals, and you can see the classic inverted U-shaped dose response curve in the adults. But the main point that I want to make here is that curve is literally lifted in the adolescents, and you see condition place preference across a wider range of nicotine doses, suggesting that nicotine is more rewarding in the adolescents. So on the other side of the arm, we can also test the aversive qualities of nicotine withdrawal. And so we take the side of the compartment that the animal does not prefer, and we give them a nicotine pump to first make them dependent, and we give them injections of mecamylamine, a nicotine receptor antagonist, to precipitate withdrawal in the presence of those environmental cues. And so in this case, values below 0 reflect the aversive qualities of nicotine withdrawal. And so I'll point out that the adult animals, we saw a very large avoidance of the compartment that was paired with withdrawal, but the adolescents were insensitive to that effect. And one of the things that we did is that we even took a group of adolescents and gave them an extra 7 days of nicotine to sort of enhance their dependence measure. And we still saw that condition place aversion was lower in the adolescents. Now, one of the things we can also measure during withdrawal is physical signs. So they emit behaviors like an excess of blinking, writhing, gasping, body shakes, ptosis, which is basically closed eyes, grooming and licking behavior. And what we saw in these data is that adults show a nice, so the black bars are the adult animals that were exposed to nicotine, and when we give these animals mecamylamine, we see an increase in the somatic signs of withdrawal, but these effects are lower in adolescents. And so we replicated these data with equivalent doses of nicotine across lots of different doses of mecamolamine, and still continued to see that withdrawal was lower in the adolescent animals. And so I put this data in a little bit after I saw Dr. Fowler's amazing presentation, because we can also take advantage of these models and expose animals to nicotine and test their propensity for rewarding effects later on in development. And so this study was a self-administration study where we compared adolescent and adult animals and looked at their nicotine intake in a model of extended access to nicotine self-administration. And we let the animals press for nicotine in 3-week increments of time with increasing doses of nicotine, and then we repeated that in a second cycle. So on the left, you're looking at adolescent rats actually as they age. And so what you can see is that the adolescent animals press a lot for nicotine, and you can compare them to naive adults on the right, which is the solid squares. So adolescents are pressing a whole lot more for nicotine than the adults are. But probably the coolest group to point out to you is that the animals that are pre-exposed to nicotine during adolescence and were tested as adults also show an increase in self-administration. So those are those black triangles compared to the black squares. So it seems that even being exposed to nicotine as an adolescent enhances the rewarding effects of nicotine later in development. So to summarize, I'm showing you that the rewarding effects of nicotine are greater during adolescence, but the withdrawal signs are lower, both physical signs and negative affective states. And so the next series of studies we did were looking at the neurochemical mechanisms of these behavioral effects. And so we wanted to, obviously there's a lot of different brain regions that we could have begun to do this work, but we focused on the nucleus accumbens. And the reason is that synaptic levels of dopamine are decreased in the nucleus accumbens during withdrawal. So they're increased by nicotine itself, but they're decreased during withdrawal. And so this phenotype, if you'll allow me to call it a phenotype of dopamine, is something that we use to begin to ask questions about development. And so our question was, are the decreases in dopamine in the nucleus accumbens similar across age? So we used in vivo microdialysis. If you're not familiar with this method, it's a dialysis probe that's lowered into a brain region. And you put artificial cerebral spinal fluid into the fluid that runs through the probe, and by diffusion, the neurotransmitter seeps into the dialysate, which then you can measure using different quantitative methods. We use mass spec to be able to look at an array of neurotransmitters. And so in this study, the animals were first implanted with a nicotine pump, and then we put a probe into their brain, and we assessed changes in dopamine levels during precipitated nicotine withdrawal. And so I'm going to back up just for a second to give you a little bit of background to make sure we're all together in terms of the mechanisms. As we know, dopamine is increased in the nucleus accumbens during after nicotine administration, and it's believed that dopamine plays a primary role in modulating reinforcing effects. Now, what's been shown is that when nicotine is placed, this is work largely by Mansfelder and John Danny, showing that when nicotine activates alpha-7 receptors, those receptors hypersensitize. And so you have an excess glutamate that's released into the ventral tegmental area, contributing to the increase in dopamine. On the other hand, the nicotine binding to alpha-4 beta-2 receptors, those receptors desensitize in the presence of nicotine, so you have removal of an inhibitory neurotransmitter. Both of these things work together to increase dopamine in the accumbens. During withdrawal, this has been quite some time ago, but we had some evidence in the literature that during cocaine withdrawal, there was a decrease in dopamine in the nucleus accumbens, and we hypothesized that that decrease in dopamine would be due to less glutamate and greater GABAergic inhibition of the dopamine pathway. And so in this first study, again, we had adolescent and adult animals that had a nicotine pump, and we measured dopamine levels during a baseline period and then following administration of mecamolamine. And what these data are showing is that across time, you see that the dopamine levels are decreased in the nucleus accumbens of adult animals that are experiencing nicotine withdrawal. However, the adolescents were insensitive to this effect. Then we wanted to look at GABA, and this was a separate study where we looked at the release of GABA back in the ventral tegmental area. And what we observed is that in adult animals, we saw an increase in GABA release in the VTA, and again, the adolescents were not sensitive to this effect. And then the last neurotransmitter that we looked at was glutamate, and what you can see is that the adult animals display a decrease in in glutamate in the VTA, so greater inhibition, reduced excitation. And so just to bring back the hypothesis that we had talked about, so during withdrawal, we saw the decrease in dopamine in the male rats, and this seemed to be due to amino acid regulation of glutamate and dopamine. And so in the adolescent animals, we saw this insensitivity to the decrease in dopamine. And the reason we're hypothesizing that the adolescents are insensitive to that is that they have greater glutamatergic inputs to the nucleus accumbens. And also, if you look at GABAergic IPSPs in the adolescent rat, they're lowered. And so it's less inhibition and greater excitation that we argued was protecting the adolescents from the decrease in dopamine in the accumbens. Okay, so to summarize the behavioral results, I showed you that the rewarding effects of nicotine are greater in adolescents, but withdrawal was lower. I also showed you that adults show a profound decrease in dopamine in the accumbens, but the adolescents were insensitive to this effect during withdrawal. And so the take-home message is greater reward, lower withdrawal during the adolescent period. And I'm going to bring you back to another diagram that my student showed where nicotine is basically a very strong on switch in the brain of adolescents. And so the idea is, and the possible clinical implication is that the very strong rewarding effects of nicotine during adolescence likely drive use. It could be the case also that nicotine facilitates the use of other drugs or social interaction or risk-taking behavior that's characteristic of that young period of development. And so this is silly for a rat person to say in front of some clinical people, but I think what our data suggests is that avoidance strategies are really key to reducing nicotine use in adolescents. And in terms of withdrawal, I think what our data suggests is that we really need to think very carefully about the diagnostic criteria that we use for adolescents because they were developed for adults. And it's also the case that treatments that reduce dopamine or fixing the dopamine deficits that you see in adults may be less effective in adolescents as compared to adults. And there's some work by Eric Mulchan and others showing that it does seem to be the case that medications that facilitate dopamine may be less effective in adolescents. So switching gears to the second part of my talk, which is to focus on females. And unfortunately, females are also more vulnerable to using nicotine. If you look at the clinical literature, women have lower cessation rates and are less likely to benefit from treatments such as nicotine replacement therapy as compared to males. And during withdrawal, females experience really high levels of depression and anxiety, and they'll report that the anxiety-reducing effects of nicotine are one of the main reasons that they continue using. And so this, and also I should mention that the general smoking rates are going down, but there seems to be a persistent increase in females, particularly in young females. And so our work more recently has been focused on the role of stress in particular in contributing to nicotine use in females. And so just a commentary a little bit, as I did with the developmental piece. For studying sex differences, it's quite easy. We include male and female animals in our work. But I'd like to suggest, and really just have a teaching moment, that when you start to look at ovarian hormones, this is a separate question. These are not interdependent questions. But when you begin to look at ovarian hormones, there's lots of different manipulations and ways in which you can study the role of ovarian hormones in modulating the behavioral effects of nicotine. And so some of the ways that we do that in our animals is by ovarectomy, which means you remove the ovaries and remove one of the major sources, or the major source of the production of ovarian hormones. We've also started to begin to measure hormone levels during key test days to see what the relative levels of the hormones predict about the behavioral outcomes. And we've also started working with different paradigms to remove and then replace ovarian hormones, which is very challenging. And so I put this slide just to show you that obviously the female cycle is 28 days, but in the rat it's 4 days, but it has the same characteristic fluctuations in hormones. And so this is a nice model to also study the role of ovarian hormones. And so the first study that we did to compare sex differences in the behavioral effects of nicotine utilized a 23-hour access to self-administration of nicotine. One of the nice advantages of this procedure is that we can also measure food and water intake, and we can look at diurnal patterns and all different sorts of variables that could impact nicotine intake in males and females. And so we had groups of animals, males and females, and then we included animals that had received an ovarectomy. And then we had groups of animals that received estrogen supplementation after they had been overectomized, and we used different doses of estradiol in that replacement procedure. And so the data, this is part of the data, comparing the males and the females. The females are in red, and you can see that they had displayed significantly more nicotine intake relative to the males. And probably the neatest part of this data is that the overectomized animals showed a reduction in nicotine intake that looked very much like the male phenotype. And so this is another kind of neat aspect of this project where we took the overectomized animals, we had different groups that had their estrogen supplemented, and we see, at least at very high doses of estrogen supplementation, we saw an increase in nicotine self-administration. And so this suggests that estradiol in the rats is likely a key hormone that promotes its rewarding effects. So as we were digging into the literature, I had a wonderful graduate student, Rodolfo Flores, who's now at NIMH doing his postdoc. But Rodolfo was taking a meta-analysis course, and he said, you know, this literature isn't as clean as we had suspected, which is sometimes the case. May I do a meta-analysis? And I said, sure. And it turned out to be a wonderful experience because it meant I got to connect with all of these colleagues that had done male-female nicotine self-administration to beg them for their effect sizes. And so we did an analysis where we compared effect sizes across all of the existing papers that had looked at male and female self-administration. And so to simplify, I'll just tell you that if the point is on the right, that means that there was a greater nicotine intake in a female versus a male. So you can sort of just go down the row, so hopefully some of the authors are here in this audience, to see that in general, when we collapsed and looked at our combined fixed effects, in general, females take more nicotine than the males. So one of the other neat things about meta-analysis that I learned is that you can look at moderator variables because, of course, every study across every different lab had so many different variables that we worried would impact our overall analysis. And so we looked at a bunch of different things, including age, cues, whether or not they looked at the night or the daytime. And we found 4 variables to emerge as really important. And so if you're interested in doing this work, of course, age matters. You can see that the adult animals take more than the adult females take more than the adult males, and this effect is smaller during adolescence, probably because of the role of ovarian hormones. Cues seem to play a role, but also under different schedules of reinforcement. Females press more than males under really tougher schedules of reinforcement. And then lastly, we saw very significant effects when the animals were given extended access. So to summarize, I've shown you that the rewarding effects of nicotine are greater in female rats, and this effect is obliterated by overectomy procedures. And it's normalized to intact female levels when you replace estradiol. And so we also learned that there's key procedural variables, including age, cues, and the schedule of reinforcement that can magnify the sex differences. And so we wanted to ask about whether or not there was sex differences during nicotine withdrawal. And we did a study where the animals were implanted with a nicotine pump for 14 days. And then we tested them 24 hours later into withdrawal. And we looked at anxiety-like behavior, but we also took their brains and looked at changes in gene expression of various genes that we had hypothesized would play an important role. And if you're not familiar with the elevated plus maze, rats have a natural tendency to avoid open spaces. And so in the elevated plus maze, if an animal spends more time on an open arm, that means it is, more time on the closed arm means that it's more stressed. I'm saying that because that's how I show the data. So we can use this increase in closed arm time as an assessment of anxiety in the rats. So these are our data. On the left, I'm showing our measures of anxiety-like behavior, and on the right, they're measures of plasma corticosterone levels. And we'll start by talking about anxiety-like behavior, which was increased in the females versus the males. And so that's the black bar with the asterisk above it. So very much of a stronger behavioral response in the females versus the males. But we saw that when we took their blood, when we were done with testing, the plasma corticosterone levels were much higher in the females versus the males. And so the behavior and the biological measures seem to be in line with one another, that the anxiety and the biomarkers were in parallel. And so when we started looking at where these effects might be modulated, we chose to look at two terminal regions of the mesolimbic pathway, the nucleus accumbens, and also the amygdala, because Olivier George had shown that CRF in the amygdala was enhanced during withdrawal in male animals. And so we had nice reasons to look at those two brain structures. And so on the left, I'm showing you gene expression of CRF in the nucleus accumbens, and on the right is the amygdala. And we had to add a key control group because we wanted to make sure that we controlled for the effects of nicotine itself. And so in red are animals that just received nicotine, and they were sacrificed with nicotine still being delivered so that we could differentiate the effects of nicotine from withdrawal itself. So on the left, what I'm showing is that the magnitude of CRF gene expression was higher in the nucleus accumbens of females versus males. And what I love the most about this data is it's separate from the effects of nicotine exposure itself. And then on the right, I'm showing that in the amygdala, much like we might have predicted from the existing literature, the gene expression in the amygdala was higher in the males. And so it seems to be lower in the females. So the coolest part of this data is that it's region-specific and sex-dependent. So to follow up on this work, we did another study where we included a group of animals that were either intact or overectomized. And we had animals that were control animals that were not exposed to nicotine. We included the nicotine-exposed animals. And then we had animals that were tested in a state of withdrawal. So on the top left, you can see the replication of the finding, which is that adult females show a large increase in anxiety-like behavior that's higher than the males. And on the bottom left, I'm showing you that we see similar increases in CRF gene expression, but we added a new gene, which is urocortin, which was also higher in the females than the males. And so in a way to sort of wonder, this is behavioral neuroscience is a correlative field to some extent, but how can we look at how these things are related to each other? And so we tried correlating the magnitude of anxiety-like behavior with the degree of gene expression. And what we found was that the more urocortin gene expression the intact females had, the more anxiety-like behavior that they exhibited. And so these things seem to be very related to nicotine during a state of nicotine withdrawal. And so to summarize, I've showed that females show greater anxiety-like behavior and biological measures of stress during withdrawal. And the intense stress appears to be mediated by stress-associated genes in the nucleus accumbens. These effects are absent. All of these effects are obliterated by ovarectomy procedures, suggesting again that ovarian hormones are modulating the strong effects of nicotine in females. And so the next series of studies that we did were asking about what are the mechanisms of these effects. And so we looked at the nucleus accumbens in large part because we had already learned that the differences in terms of age were modulated in the nucleus accumbens, and so we went to look at sex differences in the release of dopamine in the nucleus accumbens. Now, one thing I'll point out here is just focus a little bit on the fact that dopamine release in the nucleus accumbens is under inhibitory local control by GABA. And so not only did we measure dopamine, but we measured GABA release in the nucleus accumbens. These are male and female animals that had a nicotine pump, very similar. They had a probe, and we gave them mecamylamine to precipitate withdrawal while they were being assessed for changes in dopamine and GABA. So these are the data that we showed, and so on the left I'm showing the release of dopamine during withdrawal, and you can see that both the males and females show a decrease in dopamine in the nucleus accumbens, but the decrease was larger in the females than it was in the males. And I will say that the previous, this was done with mass spec because these are very, very, very low levels of dopamine that we were trying to pick up on. So all of these data are now done with mass spec procedures. And another interesting thing that we learned is that GABA levels were increased, but the increase was higher in the females versus the males. So what's going on? We learned that the males show a decrease in dopamine during withdrawal. GABA levels are increased. But the main point of this data is that the GABA release is higher in the females. And this is admittedly a hypothesis for us that you have greater regulation of the nucleus accumbens by CRF, because we did see also an increase in CRF gene expression, and those CRF receptors are likely there to enhance GABA levels, contributing to the large decrease in dopamine that we see in the nucleus accumbens. And so ongoing work in our lab is trying to figure out how these players come together. We really wish we could measure CRF in dialysis, but there's lots of challenges associated with that. So to summarize, I've showed you that females show very, very strong rewarding effects of nicotine, but also very, very strong withdrawal symptoms, and more specifically in the realm of anxiety. And our biomarker analyses showed you that we saw a greater release of stress hormones and changes in stress-associated genes in the nucleus accumbens than males. And so all of our effects, again, were modulated by ovarian hormones. So the take-home message here is both reward and withdrawal are enhanced in females versus males. So I wanted to make a few clinical implications again for the females, and so the idea here, this is another diagram that was drawn by my student, showing that the strong rewarding effects of nicotine likely drive use in females, but it's really stress and anxiety that has a profound grip and role, probably contributing to the high relapse rates in women. And so we would like to suggest that stress is a really critical factor in terms of treating women for smoking cessation and nicotine use. So I wanted to touch a little bit about some recent exciting work that we're doing, sort of expanding beyond the mesolimbic pathway, beginning to look at other pathways. And recent work has shown that the projection from the medial habenula to the IPN plays a very significant role in modulating the aversive aspects of withdrawal. And we began to collaborate with Mariela De Biasi, who was one of the leaders in sort of discovering the role of this pathway in modulating nicotine withdrawal. And so we've done a series of studies more looking at IPN, showing that the IPN is a structure where we see profound differences as compared to males. So we see greater release of GABA, we see greater upregulation of alpha-5 subunits. And I'm showing you on the right some data that sort of just gets me the most excited, which is the just general FOS expression is higher in the in the females than it is in the males. And so that's the red bar. You see the females showing really, really high levels of FOS expression, mostly localized to the central portion of the IPN. And this very nice large expression of FOS was reduced by a CRF1 antagonist, suggesting that those effects of inhibitory tone in the IPN are likely modulated by CRF1 receptors. And so Dr. Fowler nicely pointed out that we're starting to look at models that we believe will more closely mimic human nicotine use. And so we've moved to nicotine vapor methods, and this is a study that was just accepted for publication where we videoed the animals. And so we had adolescents, adults, males and females, and we exposed them to nicotine vapor. And one of my wonderful and astute undergraduates noticed that by the end of the study, the adolescent animals, in particular the adolescent females, were sticking their nose inside of the port where the nicotine was being delivered. So I said, okay, let's do a microanalysis of this behavioral effect, and sure enough, we saw that, and this is the black bar in the middle, the adolescent females showed significantly greater increases in nose poke behaviors by the end of the exposure procedure. And we're hopefully going to capitalize on this effect by allowing the animals to then perform additional nose poke responses to then begin to look at volitional aspects of nicotine. So they'll be able to self-administer nicotine vapor in the next series of studies that we conduct. So I'm going to switch gears a little bit. There was an opportunity at UTEP, being from a border region, to begin to look at health disparities and things that affect different populations of people. And so they funded a small project from a student that I had that was very interested in studying diabetes. And so we took this on in large part because diabetes is a major public health problem, particularly in my city where I live. And we noticed that, first of all, just from a clinical perspective, nicotine use and diabetes together produce comorbid health issues. So each of these use of nicotine and diabetes can make health outcomes significantly worse. And so I think this is an important population to study. Now, these bullet points are simplified from the clinical literature, but there is evidence that persons with diabetes may be more vulnerable to using nicotine. So diabetic persons report greater pleasurable effects of nicotine. They are also less likely to quit smoking, and they're also more concerned about gaining weight if they quit. And they also, interestingly, display greater anxiety during withdrawal. And so these clinical findings really inspired us to begin to look at what are the neurochemical mechanisms of these effects. And so this is a long process that we engaged in to begin to study diabetes in rodents, which is not easy. They're not intended to be metabolically challenged. But just in the way of background, insulin is a hormone that's released from the pancreas and takes up glucose in cells. And so there are different types of diabetes. If you're not familiar, type 1 diabetes is a deficit in the release of pancreatic insulin. And it's typically associated with adolescence because the onset in persons with type 1 diabetes occurs during adolescence. And the rodent model that we've been using to try to model this form of diabetes is using streptozotocin, which is a drug that basically destroys insulin-producing cells. And so it produces a reduction in insulin and an increase in glucose levels. For trying to model type 2 diabetes is associated with obesity, and the rodent model that we use is chronic high-fat feeding. And so both models produce a long-lasting decrease in insulin signaling and an increase in plasma glucose levels. And so we've used these models to ask really exciting questions about what the role is of insulin in modulating the behavioral effects of nicotine. And so this was a study that we did. The first study that we did was just using place conditioning that I've already explained to you. On the left, we looked at conditioned place preference, and on the right are animals that had a nicotine pump, and we precipitated withdrawal in the conditioned place aversion procedure. And on the left, what I'm showing you is that you see the inverted U-shaped dose-response curve in the adult controls. But animals that received STZ displayed higher shifts in preference. So this suggested that the rewarding effects of nicotine were higher in animals that had received streptozotocin. On the right, I'm showing you that we did see that nicotine withdrawal produced aversive effects, but the withdrawal response was significantly higher in animals that had received STZ. And so we wanted to, this whole process has been about different models and trying to get at the question from different angles. And so we used extended access to self-administration to see if animals that received STZ would show differences in nicotine intake. And this is where this extended access model was so key for us, because we've been able to look at food and water, but also presses and nicotine. And so the food and water has served as a nice indication that our animals are in fact metabolically challenged. And so this is a really simple slide just to show you that STZ produces a profound increase in glucose levels, but that our insulin, we use insulin pellets, our insulin pellets normalize the glucose levels in our model. And so this is the coolest part of the data, where we showed that the red squares are animals that had received STZ. And you can see that these animals display a robust increase in nicotine intake compared to all other animals. And perhaps the neatest part of the data is that the black squares reflect animals that had received insulin supplementation. So we saw an increase in nicotine intake that was normalized to control levels following insulin supplementation. So admittedly, the STZ model is messy. This is a drug that gets taken up by glucose transporters, and those are not just located in the pancreas. They're located all over the body, and so we had to conduct a lot of work showing that our effects were not likely due to other side effects of this medication. And so what we decided to do is begin to ask whether or not under other conditions of hypoinsulinemia, can we see the same behavioral phenotype? So these are animals that are self-administering nicotine again, but before they went into that, they received a chronic high-fat diet regimen. So they received 12 weeks of a chronic high-fat diet regimen. And so on the next slide, I'll show you the data. So the animals received the chronic high-fat diet, and if you look at the data on the top, you can see that we saw absolutely no difference. And we also learned that these animals were not insulin resistant. And so this is a problem, because this is what we wanted to study. And we learned later that it takes about 20 weeks of high-fat diet, extremely expensive, extremely timely, to look at these effects. You can eventually see insulin resistance with a high-fat diet. But what we did is, in a separate cohort of animals, we had them feed for same amount of time, but in this case, we gave them a low dose of streptozotocin to sort of shoot them over the edge to insulin resistance. And this is the really neatest part of the data, is that you can see that these animals on the bottom that were insulin resistant show the phenotype of greater nicotine intake. And the neatest part of the data is that we compared males and females. And on the left, you can see that the females showed an even greater increase in nicotine intake as compared to the males. So to summarize, I've shown you that hypoinsulinemia produced by STZ increased the rewarding effects of nicotine and also withdrawal from this drug. The increase in nicotine intake was normalized following insulin supplementation, suggesting that insulin plays a role. Insulin resistance, also produced by a high-fat diet, did increase nicotine intake, but it meant that they had to be insulin resistant, and this effect was larger in the females versus the males. And so the take-home message is that both nicotine reward and withdrawal are enhanced in rodent models of type 1 and type 2 diabetes. And so here's another diagram. I really love this diagram because it shows all of the different reasons that someone suffering with a metabolic disorder may smoke. And so hopefully you can see it from where you're sitting, but insulin regulation, weight gain, lots of different factors. And I think the most exciting part of our data in a rat, because I always say a rat doesn't care if he's fat, is that there's a greater drive to use nicotine. And so I think that this data suggests that both rewarding effects and strong dependence both modulate nicotine use in persons suffering with diabetes. And then also, I think that this data really suggests, and this may just be like a no-duh moment, but we really have to think about very close regulation of insulin as we're also treating people for their smoking cessations. So overall, I always like to admit whenever I give this talk that when I first started doing this work, I thought it would be one mechanism. Like there'd be one reason that all of these people are vulnerable to using nicotine, and of course that's terribly naive. But the main point, I think, of this work is that different groups of people come to smoking for different reasons. And I think those subtle differences imply that we need specialized interventions. And so this diagram on the right is from a review paper where we took the Kubian downward spiral and sort of made it different for these different populations that we study. And you'll notice across all of the different populations that we study, each of those show greater rewarding effects of nicotine. And so that seems to be a consistent phenotype across the vulnerable populations that we've studied in rats. But in terms of withdrawal, we see a little bit of a difference there where the persons with diabetes and females show strong withdrawal, but not in the adolescents as much. So this suggests, especially as we treat withdrawal, that there may be subtle differences in specialized medicines for these populations. So I'd like to wrap up my talk by just sort of maybe plugging, silly to plug it, but I just have been working on and thinking a lot about what are the different factors, and this has been such a wonderful meeting to learn about, all the different reasons that people come to use nicotine and tobacco products. And I was given the opportunity, I'm calling it an opportunity now, at the time it didn't feel like an opportunity, but I was given the opportunity to coordinate a special issue for neuropharmacology to ask different colleagues in the field, what do you think are the different factors that contribute to nicotine use? And so I approached my colleague and friend Arbi Nazarian, and we guest edited a special issue together. And we did this front cover figure where we're showing these are all the different factors that we believe play a role. And those include sex, gender, age. We also had papers on impulsivity, different social factors. We even had somebody talking about incarceration rates. And it really gave me a nice opportunity to learn pain and other factors that are really critical for driving nicotine use. And we asked George Koob to write the introduction with us, and he allowed us to take his allostatic model and show that there are different weights in different vulnerable populations that sort of increase the allostatic load and drive nicotine use, again, across different vulnerable populations. And so this wraps up my presentation. I just want to thank you again for inviting me to come, and I want to acknowledge NIDA and the American Diabetes Association who funded this work. I'm super grateful for the opportunity to be a mentor to trainees and to help them and see them move along their paths. And this is a picture of my lab right before the pandemic. So thank you so much, and I'll take questions if you have any.
Thank you, Laura. That was a really amazing talk, and it makes me so glad that you said yes when I asked you to come. So, question.
Hi, Ken Perkins, University of Pittsburgh. I just have a question about the translation from preclinical to clinical for the sex differences, because we've pretty consistently shown or seen less sensitivity to nicotine dose manipulations when it comes to reinforcement of reward in women compared to men. And yesterday I showed a slide with our meta-analysis of patch in women versus men for nicotine versus placebo, with women showing about half the efficacy for cessation compared to men. It's like a little under 15% versus 10% for women, nicotine to placebo versus 20% versus 11% for men. So I'm just wondering about the translation, how complete it might be for this particular the rodent and the human, and exactly what the measures are that are being compared. We do not see across the board differences in sex differences in nicotine, because that would be a pharmacokinetic effect, basically.
Yeah.
So that would not. So this would be it would be not that exactly. It's just a responsiveness to it.
Yeah.
Pharmacodynamic effect, basically.
Yeah. Thank you for your question, Dr. Perkins. I think that's a question I would actually ask you, because I think part of the challenge is figuring out, when we have a rat, they haven't used nicotine before. And so we have clean subjects and we're looking at sensitivity. We're also learning that once an animal is dependent on nicotine, sometimes those phenotypes can shift. And so, it may be that different levels of dependence are modulating those aversive effects. And I will tell you, when we first, and I think it's also the aversive qualities and the lethal qualities of nicotine. So we had used the same dose regimen for years, 0.01, 0.03, 0.06. When we started doing the female work, we opened the cages and found that a couple of the females had self-administered such high doses of nicotine that they died. We had never seen that before. This is a naive animal that was just starting out. So that ability to titrate, I think, is different starting out, but I think it changes likely very differently as somebody continues to use. And probably the neatest aspect of this that we're starting to see is that it's probably very much driven by the fluctuations in ovarian hormones. So teasing it all apart, I think, is very, very complicated. Probably the neatest finding that we've had more recently is that animals that had higher levels of estrogen had a stronger withdrawal severity. The animals with higher levels of progesterone relative to estrogen had lower withdrawal severity. So even when we figure out these basic sex differences, we're going to have to look across the cycle. It's probably going to be very different in aged women who are in menopause and no longer have the regulation of these hormones. So apologies if that's like a really softball answer to your question, but I do think that in terms of females, it's going to be very tightly regulated by hormones. I'm a fan, by the way. Thank you for your question. Very nice.
I also should have said it was an excellent presentation, very comprehensive. I should have started with that, which I intended to.
Okay.
I just got right into the question, so sorry. It was a great presentation.
I feel the love. Thank you. Thank you. Suchitra.
Suchitra Krishnan Saran, Yale University. I'm going to start out by saying, yes, here's lots of love.
Thank you.
That was an excellent, outstanding presentation. I always learn so much from you, Laura, every time you speak. So thank you for that. You actually got me thinking a little bit more, and a lot of the work that I do is also on developing interventions for youth, for adolescents. So with the model that you presented, it's a very interesting perspective and fits with a lot of the clinical data, which suggests that medications like nicotine patch and so on and so forth don't work that well in youth. And it fits perfectly with what you showed us today, that they are very sensitive to the rewarding effects of nicotine, but maybe what the medications we are using to treat them should not be targeted towards withdrawal. So I have two questions about that. What are your thoughts about how we target this extra sensitivity of reward in adolescence? Do you have any ideas about how you could do that? And then second question that I have for you is it possible that the models of withdrawal you're using are a little bit more of the traditional models, which look at, things like ptosis and everything else that you described, but that they might be these anxiety-related behaviors which are underlying the addiction process in adolescence? So, sorry, lots of questions. So.
No. You may have to help me remember all of it. And I want to thank you. Suchitra was one of the first ones that came up to me at SRNT, and I was scared to talk to her. But thank you so much for always supporting me. And it is really nice to see that your clinical adolescent work fits so nicely with the rodent work. I think that's really exciting. Your comment about sensitivity to nicotine, if we're going to put patches on young people, we've got to really think about the long-term changes that might happen. I'll give you a good example. The adolescent animals that we give a pump to, we measure their weight, of course, as they develop. And what we find is that the weight suppressant effects of nicotine, they're tolerant to that, having been exposed to nicotine as adolescent. So you have these young females that might take nicotine to suppress their weight, but then they're tolerant to that later in time. So I think, I don't know what the answer is. It may be that if we use a patch, there should be different doses. I'm here with my daughter, and we see a lot of adolescents putting patches on their body before they go out at night on TikTok, right? And so I think that the adolescents have figured out that this is a really nice tool. And so it's really scary, and I don't have answers for that. And your second question was about...
The withdrawal symptoms.
Great. So this gives me a chance to elevate some of the recent work that we've been looking at, where, we used to just look at the physical signs of withdrawal and put a total, just as I did. But there's really elegant studies, some I saw at this meeting, showing that those different behaviors mean different things. And there's sex differences in grooming, but not licking, and ptosis. And so even just the behavioral expression of withdrawal in physical signs, Olivier George and others, Christy Fowler, are beginning to teach us what those different physical signs may mean. As we look at the IPN, we are also seeing some very different correlations with physical signs versus affective states. And that's fascinating to imagine that the physical aspects of withdrawal are completely mechanistically separable from the affective states. It makes sense, but we're just beginning to figure that out in our rodent models. And so you're right, these are traditional, we're using the same tools, but I'm really excited by my colleagues that are looking at the microanalyses of these different behaviors.
Thank you.
Give us a little time, right?
Yeah. And money, yeah.
Time and money, thank you, Christy.
Definitely. Hi, I'm Megan Piper from the University of Wisconsin, and once again, fantastic talk. As a clinical researcher, I found this so fascinating and easy to understand, so thank you for that.
Awesome.
And one question I have for you, as I was thinking about your models, we're starting to think about what are the mechanistic reasons for other vulnerable populations to be suffering more from tobacco dependence, trouble quitting, that sort of thing. And, chronic stress has definitely been proposed in terms of, stress related to discrimination, stress related to early adverse events in childhood, that sort of thing. Is there a way, or can you conceptualize a way that your rodent models would be able to address chronic stress and then do these same mechanisms to support that notion or study those hypotheses?
Thank you so much for the compliment and for the question. So I think, part of the reason I love being a rodent researcher is that we have these things at our fingertips to be able to study. I will be honest to say that the diabetes work has nearly killed me because it is really difficult to take apart all of the different factors when you're working with rodent models. For the females, it's the hormones and the fluctuations and the feed and the caging. With the diabetic animals, there's neuropathic pain. There's a lot of different components that we also have to pay close attention to. Now, that doesn't stop me from being excited about questions like chronic stress. We need to look at what chronic stress does in these models. I'm also very interested in looking at rodent models of schizophrenia. There is no greater vulnerable population than someone suffering from schizophrenia. I think the smoking rates, Suchitra could help me, are 99%. And so there are populations where we should see some of these phenotypes hold up. We haven't done that yet. But those are the types of directions that I hope others in the field and we're certainly thinking about also in terms of trying to figure out what it is. For now, I mean, my guess would be that chronic stress, because there's a hypothesis that CRF is more released during nicotine exposure, would just elevate that. So chronic stress would even create a greater regulation of the mesolimbic reward pathway. And so my guess would be that it would be even worse.
Great. Thank you again for a great presentation.
Sure.
Okay. Any other questions? I think we're over time, so we will call it there. And thank you, everybody, for coming. Thank you, Laura, again, for an amazing talk.